All right, everybody. So hopefully you can still hear me. Just changed my microphone. So here we have the most recent announcement. Let me know, I said we could try this idea of starting at 6 a little bit earlier, but a couple of you have said that you like it at 6:30, and we can easily move back to that. I'm kind of doing it for the next couple times just to get through some material. One thing that I did want to point out to you that I totally forgot to mention is that Tuesday is Flex Day, September 21st. And technically, we're not allowed to have any sort of synchronous classes. So you guys will have the day off, but what that'll do is that'll kind of push us back one day. Remember, we're going to have our lecture exam next Thursday. So what we'll do is you'll have Tuesday off, extra time to study. We'll get through chapter 3 tonight. And then on Thursday, the 23rd, we'll get through chapter 4. And then this is important, your lecture exam will be on Tuesday, September 28th. And that'll start at 6:30 at normal class time. So you don't have to log into Zoom for that. You'll just go into Canvas. And in Canvas, the lecture exam will be open right at 6:30. And then you'll have 90 minutes to take it. And I'll leave it open for about 2 hours for that. Or however much time you need if you're a DSBS student as well. And there's a link to the lecture 1 exam that takes you to that, and you'll see the updated time and date that opens. So I'm going to jump right in to where we ended in chapter 3. So remember, there's 2 parts to chapter 3. The first part kind of went through just the basics of the cell. I'm going to end quickly chapter 3 here because I don't cover it a lot on the lecture exam. Where we ended off was with talking about resting membrane potential. And resting membrane potential is the electrical potential energy produced by the separation of oppositely charged particles across the plasma membrane in all cells. This difference is referred to as a voltage, and cells that have a charge are said to be polarized. And resting membrane potential is going to come back to haunt you when we talk about your nervous system and the muscle system. So I just wanted to bring it to your attention so you've heard it once before. The voltage only occurs at the membrane surface, and membrane voltages range from about -50 to -100 millivolts. And the negative sign indicates that the inside of the cell is more negative relative to the outside. Potassium is the key player in resting membrane potential. What that means is that the electrochemical gradient, so the amount of potassium ions and the charge of potassium, will kind of set the resting membrane potential. Most cells have this voltage or resting membrane potential of about -90. And you'll see sodium also affects the resting membrane potential. And I'm just going to show you this kind of picture that describes the key players in resting membrane potential. And again, I don't ask you guys, I don't think many or if any questions about this on your lecture exam, but I want to cover it because we'll talk a lot about it in the nervous system and muscular system. So here we have potassium diffusing down the steep concentration gradient. So it's leaving the cell via leakage channels, and the loss of potassium, which is a positive charge, results in a negative charge on the inner plasma membrane surface. So what this just means is the resting membrane potential means that at rest, when a cell is not being stimulated, like in the nervous system, or contracting, such as in the muscle system, the inside of the cell, right on the inside of the plasma membrane, is more negative. When the cell becomes more positive, that means it's being kind of fired or ready to move. And it's positive on the inside because of potassium leaking out and because of these protein anions, which are very negative. So that's kind of this resting membrane potential. Active transport maintains the electrical chemical gradient. So we were talking about the sodium potassium pump, and that, remember, transports 3 potassium out of the cell and 3 sodium out of the cell and 2 potassium into the cell. And that kind of active transport pump will establish the concentration of these ions on the inside and the outside of the cell so that we kind of establish this resting membrane potential. When the inside of the cell is at resting, which is again at about -90 millivolts, that means it's in the best possible place to receive a stimulus or to contract, depending on if we're in the muscle or nervous system. So that's kind of, I just wanted to bring that to your attention because we will talk about that a lot. Even one of your workshops has to do with graded potential and action potential. The cell environment interactions, I want to talk a little bit about CAMs and plasma membrane receptors. This is just referring to how cells are interacting with their environment by responding directly to other cells or indirectly to extracellular chemicals. And we'll talk about cell adhesion molecules and plasma membrane receptors, which are all involving the glycocalyx, which is kind of that layer of sticky sugars on the plasma membrane. The CAMs function to anchor the cell to the extracellular matrix, assist in the movement, attract white blood cells if a place is injured, stimulate the synthesis or degradation of adhesion or adhesive membrane junctions, and transmit intracellular signals to direct cell migration, proliferation, which is kind of the cell dividing, and specialization. Some of your plasma membrane receptors will act in contact signaling so that cells can recognize each other, or chemical signaling where there's an interaction between a receptor and a ligand, which is a chemical messenger that can cause changes in what that cell will then do. So these kind of backing up here, these are really important, and we won't go into detail into these, but how your cells in your body interact with each other and how they respond to different chemicals or disease. Eventually, a chemical, a disease, a hormone gets into a cell, and it does so by some sort of adhesion molecule or a plasma membrane receptor that can recognize it. Some examples of these ligands, which are chemical messengers, can be neurotransmitters, hormones, paracrines, chemical signaling. This talks a little bit more about that, which I won't, again, ask you too many details. I do want to talk a little bit about G proteins. They indirectly cause a change in the cell by activating G proteins, which in turn can affect ion channels, activate other enzymes, or cause the release of an internal second messenger chemical, such as cyclic AMP or calcium. So G proteins kind of act as this middleman or relay person between an extracellular messenger and an intracellular second messenger. We'll talk a little bit more about G proteins later, but again, I just like to kind of bring this to your attention so you're a little familiar with it. All right, so now we're going to go on to part 2, and I'll go ahead and share my screen. So part 2 has to do a lot with the organelles in the cell. So we're still talking about a cell. On Tuesday, we'll talk about tissues, but I promised after this kind of module, we'll get more into body, kind of more interesting things. So the first part of this second part of this cell chapter is all about the cytoplasm and the cytoplasmic organelles. And you will have some sort of question about these organelles where you need to know what the organelle looks like and what its function is. And that is the case for the organelles listed here. I know you covered this a little bit in anatomy, and what you learned in anatomy or another general biology class consisting of an organelle, the general structure and function, is all you need to know for me as well. So I'm going to skip over this part that goes through these organelles. But again, you should know general function and structure of mitochondria. So for example, you might have a matching question or multiple choice question, but you should know mitochondria make ATP via aerobic respiration, and you should know generally what they look like. You might have a picture. Ribosomes create protein. That's a big one, you should know it. They're made up of subunits. Ribosomes can be free or bound to the endoplasmic reticulum. That brings us to the endoplasmic reticulum, which are flattened tubes. We can have rough or smooth endoplasmic reticulum, and that just is if they are kind of studded with ribosomes or not. So here's the endoplasmic reticulum. You'll often see the endoplasmic reticulum surrounding the nucleus because the nucleus creates these ribosomal subunits, and then those ribosomes will be attached to the rough ER, and the smooth ER is more important in creating lipids. So again, I'm going to let you study on your own, but again, generally speaking, this should be a review. You should know these organelles. You should know general structure and function. You don't have to know the specifics about how the Golgi apparatus processes and distributes newly synthesized proteins. I won't ask you specifics about that. Peroxisomes are another important organelle. They contain powerful detox substances. Lysosomes contain digestive enzymes. The endomembrane system consists of the Golgi apparatus and secretory vesicles. You should know a little bit about the cytoskeleton. And the cytoskeleton, just like our bodies have a skeleton, your cells have this cytoskeleton that just helps to support it. And there's 3 types to the cytoskeleton that you should have a very basic understanding of. I won't ask you details. Think structure, function, and then that's it. So a good way to study this might just to be, can I describe the mitochondria, the intermediate filaments in 3 words or less, or 5 words or less. And that's all you need to know, just the basics of each. So I'll let you guys kind of go through that on your own. Centrosomes and centrioles look a lot like this, and they help in cell division. So here are the centrioles. They come in pairs, and they come in pairs because they'll migrate to opposite ends of the cell in cell division. Cellular extensions usually help with movement of the cell or movement of materials across the surface. And we have cilia, flagella, microvilli. Again, knowing basics of these is enough. You don't have to know specifics of how they all work, but just knowing basically what they look like and what they do. Microvilli are really interesting. You'll see a lot of microvilli often at times in your digestive system, lining like stratified columnar epithelium in your digestive system. But where we will kind of start is at the nucleus. And we're going to start at the nucleus because I want to spend some time in this chapter to talk about protein synthesis, which is the creating of proteins from DNA. Before we do that, we need to talk about where your DNA is stored, and that is stored in the nucleus. So this is where I'm going to kind of start this part. All right, so your nucleus is the largest organelle. And again, an organelle is any kind of membrane-bound structure within a cell. The nucleus contains all of the genetic library of blueprints for the synthesis of nearly all of your cellular proteins. Your nucleus can respond to signals that will tell what kind and how many proteins that need to be synthesized. Most cells have one nucleus, but your skeletal muscle cells, some bone cells, and some liver cells are multinucleate with multinuclei, and your red blood cells are anucleate. They don't have any nucleus. The structure of the nucleus has 3 main pieces to it: the nuclear envelope, which is kind of its own kind of plasma membrane, which allows things in and out, nucleoli, and chromatin. So here's what the nucleus looks like. You can see the nuclear envelope, the chromatin condensed, and the nucleolus is kind of like the inner central brain of the nucleus itself. You can see here within the nuclear envelope, you have nuclear pores, which allow for RNA to leave, and we'll talk about that a little bit. And oftentimes you'll see rough endoplasmic reticulum surrounding the nucleus because your endoplasmic reticulum that's rough is studded with ribosomes, which are useful for making proteins, and the nucleus will create these ribosomal subunits. So here's the nuclear envelope. It is the barrier that kind of encloses the jelly-like fluid of the nucleoplasm. It's continuous with a rough ER. That's studded with ribosomes, and nuclear pores just allow for substances to pass into and out of the nucleus. So here's again what the nucleus looks like. The middle picture is kind of cool because what you're looking at there are the nuclear pore complexes, and each pore is ringed by protein particles. So kind of interesting to see there. Nucleoli are dark staining spherical bodies within the nucleus that are involved in ribosomal RNA synthesis and ribosome subunit assembly. So remember here that ribosomes, these ribosome subunits will be what make protein. And our RNA, ribosomal RNA, are what will carry the blueprints for a protein out of the nucleus and into those ribosomes, which are in somewhere in other part in the cell. Chromatin then, it consists of about 30% strands of DNA, 60% proteins that are histones, and 10% RNA. Your chromatin is arranged in fundamental units called nucleosomes, which consist of the DNA wrapped around histones. What chromatin eventually condense into are chromosomes. So right before the cell will divide, your chromatin will condense into these chromosomes, and the condensed state of chromosomes just helps to protect the fragile chromatin threads when the cell will divide. So here's chromatin and chromosome structure. Again, chromatin is your DNA. So we have kind of this DNA double helix strand kind of wound up. The DNA is wound up around these histone proteins. Chromatin looks kind of like the beads on the string structure, kind of each nucleosome bound together. And a nucleosome is usually 8 histone proteins wrapped by 2 winds of the DNA strand. And what we're getting to again, so DNA will kind of wrap around these histones to create chromatin, and then chromatin will go on to condense to form a chromosome. And you can see this chromosome here. It's kind of the typical X-like shape that you think of when you hear what a chromosome is. So here is the chromosome, and each kind of half of this X-shaped chromosome is called a chromatid. And again, chromosomes are just tightly condensed, wound up DNA and chromatin. So the cell cycle. So your DNA has to get nicely condensed into chromosomes to help with cell division, because whenever a cell divides, it needs to pass on its DNA, because the DNA needs to continue to provide the genetic code for the body and any proteins that need to be made. So before a cell dies, it needs to pass on its DNA. And to do that, it will divide these chromosomes and divide the cells. So the cell cycle, I'm hoping you might have heard of mitosis before. There's 2 major periods of the cell cycle. We have interphase and cell division, which is mitosis. Interphase is usually what the cell spends about 90% of its life in, where it grows, carry out its usual activities. So if it's a red blood cell, if it's a lysosome, every cell has a unique kind of role that it plays in your body. And most often your cells stay in this interphase stage, which is they're just kind of taking on or doing their normal roles. About 10% of the time, the cells will divide into 2, and that's called mitosis. So here's interphase. Chromatin will be uncondensed. Interphase does consist of some what we call subphases, which include the process of DNA replication to prepare for cell division. Here are the subphases of interphase: G1, S, and G2. G1 is a growth phase. Synthetic. S is where DNA will replicate itself to get ready to divide. And then G2 is just another kind of preparation subphase for cell division. So here's the cell cycle, again in green, shows that the majority of the time the cell spends in interphase, which is just normal growth and development, carrying on activities, replicating DNA, getting ready for division. There's usually what we call a checkpoint or a restriction point, so that if the cell gets to that checkpoint, they'll go on and eventually divide, but they have to get to that checkpoint first. And then here we have the mitosis or mitotic phase. And we divide mitosis into 4 phases called prophase, metaphase, anaphase, and telophase. And if you had me for anatomy, I call these phases PMAT because it's just easier to remember those phases that way. So remember PMAT, that's their order. And then cytokinesis is the last step of the mitotic phase where the cell will completely divide into 2. So we're going to not rush, but we'll go over these relatively quickly because I really want to spend time in protein synthesis, or else I could just lecture all night on the cell, which I'm sure you guys would love. But DNA replication, so prior to your cell dividing, the cell needs to make a copy of its DNA because the cell needs to pass along its DNA to the 2 daughter cells. So one cell will become 2 cells, and it needs to put the same DNA in both of its daughter cells. To do this, the DNA double helix will kind of unwind, unzip. A replication fork is the point where the strands separate. The replication bubble is the active area of replication. Each strand will act as a template for a new complementary strand, and RNA will start replication by laying down a short strand that acts as a primer. DNA polymerase is the big enzyme that kind of forms the new strand. It synthesizes both new strands at a time. It only works in one direction, so the leading strand is synthesized continuously, and the lagging strand is backwards. I'll show you a picture of that. And another enzyme called DNA ligase then splices short segments together of the lagging strand. So if you remember what an enzyme is, remember it always ends in this ASE suffix. So just keep that in mind. These are enzymes because they end in ASE. The end result is two identical daughter DNA molecules that are formed. And then during mitosis, cell division, one complete copy will be given to the new cell while one is retained in the original cell. The process is called semi-conservative replication because each new double-stranded DNA is composed of one old strand and one new strand. So what do I mean by this? So let's start with our chromosome. So this is our DNA, and let's just look at our old DNA. What we need to do is we need to turn one strand of old DNA into two strands. So that's what DNA replication is. So here is the enzyme that will unwind the double helix and expose the base pairs. So these are the base pairs being exposed right here. So normally the base pairs are bound together in the complementary base pairing, adenine with thymine, guanine with cytosine. And you can see the different colors there and how they complementary fit together. The DNA strand unzips itself. So just like a zipper, your DNA strand will unzip and separate. The replication fork is the area where hydrogen bonds between the base pairs are broken and the DNA strands will separate. And then what happens here is we have an old strand of DNA on the top and an old or a template DNA strand on the bottom. And we have a leading strand, which will create the new kind of half of DNA at the top. And we have the lagging strand, which will make the second new half of DNA at the bottom. And it's just leading and lagging means the directions that they're synthesized in, and they're synthesized in opposite directions. So that's how DNA is replicated. And then that DNA will go on and be divided by the two daughter cells after the cell will divide. So these are the cells that need to constantly replicate themselves for growth and repair purposes. Your skeletal, cardiac, muscle, nerve cells, those do not divide as efficiently. So if you have damage to those cells, they're replaced with scar tissue. This is especially true. This is why, if you have a heart attack and you damage the cells of your heart, those cells aren't readily replaced. So that's why someone with heart attacks, heart damage, can be a little scary because the damaged cells will be replaced with scar tissue. And the cells of your heart are ones you just don't want to damage because they can't really regenerate themselves. Okay, so here's the mitotic phase. The control of cell division is crucial, so cells will divide when necessary, but they do not divide unnecessarily unless it's a cancer cell, which does, and that's where cancer gets interesting. So here's the cell cycle again. Here's mitosis now. Here are the 4 phases of mitosis. Each of the 4 stages will ensure that each cell receives a full copy of the replicated DNA. Prophase can be broken into 2 parts here. The big thing about prophase is that chromatin will condense to form these chromosomes, and each chromosome and its duplicate, called a sister chromatid, are held together by a centromere. The mitotic spindle occurs, which will help to get ready to divide the chromosomes. And in late prophase, the nuclear envelope breaks up. And remember, the nuclear envelope is what surrounds the nucleus, so that'll start to dissolve so that the chromosomes can eventually be separated. So here's a look at interphase. So this is normal before mitosis. Here's early prophase and late prophase. You can see how there's no nuclear envelope anymore, and you can see how those chromosomes have really condensed and they will get ready to line up. Metaphase then is when the centromeres of the chromosomes are precisely aligned at the cell's equator, and we call that the metaphase plate. So the chromosomes now have lined up. The spindle fibers that are forming on both poles are ready to separate out those chromosomes. Anaphase is the shortest of all the phases. This is just when the chromosomes will split apart simultaneously, as shown here. So that's anaphase. And then telophase begins when the chromosome movement stops, the new nuclear membranes form, the spindles disappear, and then cytokinesis literally means cell splitting. So two daughter cells will be pinched apart. So here's kind of telophase, cytokinesis, where two new daughter cells will be formed. Here's the control of cell division with different go and stop signals. I'll let you guys read through that. Different growth factors, hormones can be a go signal, a stop signal can be if space runs out or normal cell dividing. Two groups of proteins are crucial to the cell's ability. I won't ask you guys questions about these cyclins, so I'm going to skip over that. And then checkpoints are just key events in the cell cycle where cell division processes are checked. And if anything is not correct, they're stopped until repairs are made. So this is good that if there's some sort of repair that needs to be done in the DNA, the cell will not pass this checkpoint. All right, so that's the cell cycle. And then here we are in protein synthesis. So protein synthesis is the idea that your DNA forms the blueprint or the master plan for all proteins that are created in the body. And protein synthesis is very important because it creates all the proteins in the body, and proteins are what kind of make up, if you think about hemoglobin is a protein. So anytime we talk a lot about a molecule, it's most likely a protein that came from a DNA template, so your genetic code. DNA will direct specifically the order of amino acids in a polypeptide, which is a protein. And a segment of DNA that holds the code for one polypeptide is referred to as a gene. So one code for one protein or one polypeptide is a gene. The code is determined by the specific order of nitrogen bases, and that's adenine, guanine, thymine, and cytosine. And the code always consists of what we call a triplet. So the code will always consist of 3 sequential bases, where GGC, for example, like if you have a guanine, and a cytine, that will code for an amino acid called proline, where GCC, if you have a guanine followed by 2 cytosines, that will code for a different amino acid called arginine. And each triplet specifies the code for one particular amino acid, and then each amino acid put together create a protein. Genes are composed of what we call exons, introns. Exons are a part of the gene that actually codes for amino acids, and introns are non-coding segments interspersed among exons. So then we get into RNA. So RNA is extremely important because it's the go-between molecule that links DNA to protein. So in DNA, we have the genetic code of how to make the protein, but to get from DNA to actually making a protein, RNA will copy the DNA code in the nucleus, and then that RNA will leave the nucleus, carried it into the cytoplasm to where the protein will be made in the ribosomes. All RNA is formed in the nucleus, and RNA is different from DNA in these 2 ways. And this is a good test question. How is RNA different from DNA? RNA has a different base. So instead of thymine, RNA has uracil. And instead of deoxyribose sugar, RNA has ribose sugar. So that's where the R in RNA differs from the D in DNA. The R stands for ribonucleic acid, and DNA stands for deoxyribose nucleic acid. Sorry, I keep taking a sip of my tea. My mouth is dry tonight. So there's 3 types of RNA, the messenger RNA, ribosomal RNA, and transfer RNA. And to give you a little bit of light at the end of the tunnel, I did post a link to a really great protein synthesis video on YouTube. It's done by a textbook company. So after this, it's I don't know how long it is, but it's just a great YouTube video explaining this whole process. I'd really encourage you to watch it. So messenger RNA is the first type of RNA that we'll cover. It's single-stranded, so that's another difference. RNA is single-stranded, DNA is double-stranded. And what our messenger RNA does, mRNA, this will be the code from the DNA template strand. It will be copied with complementary base pairs, resulting in one strand of messenger RNA. And this is called transcription. And you should understand well what transcription is. Transcription is copying the code from DNA to a single strand of messenger RNA. Messenger RNA maintains that triplet code or the codon from the DNA, meaning that if DNA has a code for guanine, guanine, cytosine, messenger RNA will keep that same code because it's kind of just the go-between or the messenger from DNA to protein. Ribosomal RNA, so this is the second type of RNA we'll talk about. This is just the structural component of ribosomes, which is that organelle that makes proteins. And along with transfer RNA, ribosomal RNA will help to translate the message or the code from that mRNA into a polypeptide or a protein chain. The third type of RNA is transfer RNA. It's the carrier of the amino acid. Basically, what the transfer RNA is going to do is it will take individual amino acids that contain its own specific code that will match up to the code from the messenger RNA to grow the polypeptide chain or the protein. And this is called translation then. Translation is the adding on of amino acids to a chain to form a protein using transfer RNA to carry those amino acids. So these are these two steps of protein synthesis. You should know these two steps. You should know them a little bit in detail, but even if you just know the basics, that will help you out a lot. And I'll try to keep thinking of big picture items here. So again, we're trying to create proteins, and we can create proteins in just two simple steps. Transcription is the first step. That's where our DNA genetic code is copied onto a messenger RNA strand. That happens inside the nucleus. The messenger RNA strand will then leave the nucleus to go to ribosomes, which will create the proteins. And during translation, the messenger RNA will be decoded or read to assemble those proteins. So here is just a great kind of picture of where this all occurs. Transcription occurs in the nucleus, where we take DNA double-stranded, which gives us the genetic blueprint for creating a protein. We change eventually that into a single-stranded messenger RNA because it just reads that genetic code and it's going to be the messenger. Messenger RNA leaves the nucleus through the nuclear pore and it will enter into a ribosome. So this kind of blue ribbon is the messenger RNA. This blue mRNA enters into the ribosome, and translation is reading the messenger RNA inside the ribosome and adding amino acids together to create this polypeptide or protein chain. So here's transcription. Besides knowing that, for transcription means that we're transferring the code held in the DNA gene to the complementary base sequence of messenger RNA. That's pretty much all you need to know. It's broken down into initiation, elongation, and termination, but I won't even go into that detail. However, kind of talking through these three steps might help make a little more sense. So initiation, we have an enzyme that binds to DNA to unzip it. When DNA unzips, the messenger RNA starts to complementary read the base pairs of DNA. So elongation means this M messenger RNA is reading the base pairs from a strand of the DNA that's been unwound or unzipped. And the messenger RNA that's being created, its base pairs will be complementary to that DNA strand. Termination just means there's a termination signal where the completed mRNA will know that that's enough to create that specific protein. Messenger RNA will be processed. And then translation is the step of protein synthesis where the language of nucleic acids, that base sequence, is then translated into the language of proteins. And the process involves that messenger RNA, the genetic code, transfer RNA, and ribosomes. The genetic code, there might be like one question about knowing how these base pairs are complementary to each other. And I'll talk about that in a second, but each, again, 3 base pair sequence on DNA is called a triplet or a codon. This just goes through that there's 64 possible codons, because if we have 4 bases adding uracil, cytosine, and guanine in messenger RNA, and there's 3 places each to make a codon, we have about 64 different possible codons. There are only 20 amino acids. So what this means is that some of these codons more than one codon can represent for the same amino acid. So this is how we read kind of a genetic code. So let's just say a messenger RNA strand had an A, a C, and a G as its codon. So that's an adding cytosine guanine. You'll notice that there's U's in here. The uracil is in place of the thymine because we're talking about messenger RNA now. So ACG, this is the codon for one amino acid. And to read this, the first base is adenine. So we go down here. So this is our adenine. So we're going to be in this row. The second base is a cytosine. So we're going to be in this column. So right now we've kind of narrowed it down to, it's going to be one of these. And then we can already see there's only one amino acid for it. And then the third base is a G. So ACG gives us the amino acid THR, which is abbreviated for threonine. So this is the amino acid threonine. So this kind of codon, this sequence of 3 base pairs, you can see how the different kind of variations of a codon will give you a different amino acid. And this one amino acid will go on and be bound with a peptide bond to another amino acid. Let's say the next amino acid in the chain is proline. So here's proline. So proline would have to have one of these base pair sequences. And as we add on threonine, proline, serine, cytosine, tryptamine, glycine, we're going to create a protein as we add on these amino acids one at a time. The role of the transfer RNA. So this is what a transfer RNA looks like. Your transfer RNA has what we have an anticodon on it. And what the anticodon does is it just means that it kind of reads the messenger RNA and brings on the correct amino acid that corresponds to the anticodon, which just means that it's reading the codon on the messenger RNA. So the anticodon or transfer RNA will just bring the amino acid on to create a protein in translation. Translation also occurs with initiation, elongation, and then termination. I won't ask you guys the specifics of this, but what you're seeing here is you're seeing messenger RNA kind of being slipped through these ribosomes, and growing off of each ribosome is a growing polypeptide chain, which is just your protein chain that will grow on to a completed protein or a completed polypeptide. So that's what translation is. A focus, again, is just that translation is the process in which we're taking genetic information carried from messenger RNA, and it's decoded in the ribosome to form a particular polypeptide. Polypeptide means many peptides or many amino acids. So that's just another word for a protein. And again, these are the steps of translation. I won't ask you specifics on the steps, but you should know generally what translation is, what transcription is, and how these steps create proteins. All right. The rough endoplasmic reticulum goes on to process these proteins a little bit more. I won't ask you specifics on this, so you guys can skip this part as well, but the rough endoplasmic reticulum helps to process these proteins. So this is just a really good summary, kind of going from DNA to proteins. The DNA triplets are coded to mRNA codons. The mRNA codons are base paired with tRNA, And then we have the anticodon sequence of tRNA. It's identical to DNA, except the uracil is substituted for the thymine. So here we have, this is probably just a really good way to describe the idea of protein synthesis as well, because it starts with DNA and the one kind of unwound strand, and how messenger RNA will complementary base pair with that DNA. So remember, the messenger RNA going from DNA to mRNA was transcription, because messenger RNA will be the messenger of that genetic code out of the nucleus. So you can see here how these triplets are complementary. So TAC, remember that thymine pairs with adenine. Adenine pairs normally with thymine, but since this is RNA, we'll have a uracil here, and cytosine is with guanine. So as you work your way through these triplet codons, you'll notice that each base pair will pair complementary with the base pair that it should. This mRNA then will have it will complementary kind of pair with the anticodon on a transfer RNA. And the transfer RNA is called a transfer RNA because it's transferring an amino acid. And all of these amino acids that the transfer RNAs are bringing will eventually be bound together to form a growing polypeptide chain or a protein. Here's some more roles of DNA and types of RNA, which I won't cover. We'll talk then a little bit. We'll finally end the chapter. We just have a couple slides left, and we'll talk about apoptosis, autophagy, and proteasome. So autophagy is where cells that have become obsolete or damaged need to be taken out of the system. So autophagy, remember, phagocytosis means eating. So autophagy means self-eating, and it's the process of disposing of any non-functional organelles and sweeping up cytoplasmic bits by forming autophagosomes, which can then be degraded by lysosomes. Ubiquitin proteasome pathway. I'm going to kind of skip some of this, but this just helps to get rid of damaged or misfolded proteins. Apoptosis, you might have heard of before. This is called programmed cell death. It causes certain cells to neatly self-destruct. The process begins with mitochondrial membranes leaking chemicals that will try to kind of kill off these cells. The dead cells will then shrink and be phagocytized or eaten by macrophages. We try to program cell death to cancer cells or other infected cells or old cells. So that is the end of this chapter, chapter 3. I know it was a little heavy. I'm just going to take us back before I stop the recording to make sure everyone who might have joined us late, there's like 2 of you who still want to start at 6:30, and I will take that into account. Maybe just for the next really one more class that we have, I might keep it at 6 just to make sure we have ample time to get through everything. So if you ever just want to join late, you can always watch the recording. But if a lot of you are protesting that, we can move back to 6:30. You guys we will not have lecture class on Tuesday, September 21st, which I guess is already next Tuesday due to Flex Day. So because of that, I moved your lecture exam back to the following Tuesday. So what that means is that the next time we'll have a live class is a week from tonight on Thursday, September 23rd. We'll go over chapter 4. It's the tissue chapter. It's also pretty lengthy. Also a chapter you guys probably studied a lot in anatomy. So if you want to start studying that on your own, I'd encourage you to. And then the lecture exam will be a little bit outside of the module date. We'll have it during normal class time, Tuesday, September 28th at 6:30. It'll open then. You'll notice that the second module starts technically on Monday, the 27th, but we'll kind of push that off a little bit because of Flex Day. And then in terms of what you guys have due tonight, and then I'll stop not tonight, but due this week, I think you just have one lab to do, the cell membrane and transport lab, which really goes well with what we just talked about in chapter 3. And then I'll keep you guys posted on, and I'll give you reminders about other things that are due at the end of this module. So thanks for listening. And now I'll stay on to answer any questions.